So this is Lab 8, and these are specimens that we would have looked at whole. So I'm going to walk you through them and talk about their biology and form a little bit. So we're looking at animals from Porifera, Cnidaria, and Ctenophora. So this is Grantia scypha, and it's a member of Porifera, the sponge phylum. It's a filter feeder that is sedentary for most of its life cycle, and it has free-swimming larvae called choanoflagellates. So sponges are animals that lack 3 tissue layers, and they have a relatively basic body design. Grantia exhibits the cyconoid body plan, which is the second out of the third most complex sponge body plans. It has folded canals of feeding cells that stem out from the spongiocele, which is the main cavity that's open to the environment. In sponges, body plan complexity determines how large a sponge can evolve to, generally with higher complexity working for larger sponge as the general rule. So oxygenated water from the ocean has to reach all of the tissue cells of the sponge directly. You have to have direct absorption because there is no centralized place for oxygen extraction and packaging and delivery, like how we have lungs for the extraction and then a pressurized vascular system with a pump, the heart, to deliver the usable oxygen. In sponges, the oxygen-bearing water must come into contact with all cells of the sponge. So water is drawn into the sponge from the outside through the walls and out through the spongiocele opening. These preserved Grantia on the right would probably have been clustered together in an area that has enough suspended nutrients in the water for it to filter feed on adequately. They're difficult to keep alive in captivity because they don't do great in clear water. So Metridium is a genus of plumosea anemones. They have frills of tentacles surrounding the oral disc as opposed to a thick rope-like tentacle design that you would see in the type of anemone that would house clownfish in the Pacific. They can relocate by disengaging at the pedal disc from the substrate that they're adhered to and then letting the current move into a new location and setting its pedal disc down when it decides to stay there. There's some mucus and suction involved. You look at Hydra in Lab 8 also, and those do a somersault where they will bend over their stalk, use their few tentacles that are thick relative to the stalk size, grab onto the ground, and then rotate the body over and land further away at the pedal disc. I don't think that Metridium can do that because they have thin tentacles, but they can disengage and drift with the current. So all anemones are cnidarians, and they have stinging nematocysts that are good for prey capture and predation resistance. The stinging cells both harpoon and disable prey items, such as arthropods, worms, also edible detritus. I feed mine little bits of shrimp, chicken, algae pellets containing protein. They can tell that something is food, and they'll bring it into their gastrovascular cavity. So these are not this species is not adapted for catching large prey. It also couldn't hurt a person with its sting. This gelatinous animal may look like a cnidarian, but it isn't. So Ctenophora, or the comb jelly phylum, is sister to Cnidaria on a phylogeny. It lacks cnidocyte-bearing tentacles, which is something that all cnidarians possess. The comb in the name refers to its usually radially symmetric scaffolding, and the scaffolding, the comb, helps maintain body shape. It also is a platform for cilia that flutter in tandem, creating a current that moves plankton to the mouth end. And cilia movement also generates propulsion for ctenophore itself. So the moving cilia give off an iridescent sheen when they move. I've seen it in St. Lucia. A pretty big one was at the surface near the shore, and the sun was out, and you can see the rainbows that it creates along the comb. So they can give off an iridescent sheen. It's pretty interesting. All ctenophores are marine. We have some native to this area, I find them in Long Island Sound, the ones that look like the one pictured on the right. It looks like a gelatinous grape. That one could still be alive. They lack the means to envelop and contain anything large, so it is a plankton and particulate eater in full. So the ones on the left, or stage right, are a collection of predatory ctenophores, and they have big mouth openings for starters. All ctenophores are harmless to humans. So this is vastly different from what we just looked at. This is a cnidarian, which is sister to Ctenophora. So this is Gonionomus. It's a genus within the Cnidaria phylum, and it's considered a true jellyfish. It's more closely related to anemones than it is to that ctenophore, even though it and ctenophores also have a lot of gelatinous transparent tissue and drift a bit in the ocean. It's not they're not very closely related at all. So a shared derived characteristic between anemones and this Gonionemus is the presence of stinging cnidocytes on the tentacles. So Gonionemus isn't as free-floating as the Portuguese man of war, Physalia, which we're going to look at later. It's classed among the clinging jellies because it will grab onto a surface-level substrate like a seaweed cluster rather than be at mercy of the current. That may be related to why it has short tentacles. Long tentacles could be damaged while it is clinging. So a close grip is a little bit safer. So the Gonionemus is a very active jellyfish. It's been entertaining scientists since the 19th century. Its body is conducive to an active lifestyle. The bell is a tight dome, which is better for propulsion than a disc shape. The short, dense, and even skirt of tentacles keep tight to the bell. So when the bell and the tentacle skirt billows and contracts, it concentrates the energy generated by the animal, economizing it, and has a little burst of force that is more concentrated than it would be if it had a flat body and long tentacles. So it can go further with one pulse than other forms. So scientists have been dragging them into labs for a while and shining lights at them because they found that they are responsive to the 24-hour day-night cycle. During the and it is light related. So during the day, they are down low where it's deep. At night, they come up and they go practically vertical. And then when the sun comes up, they invert and float down. And I think that while they're inverted like that, they have a further reach to grab more edible items. So this is very much a predator, and its sting is toxic to humans. Populations that have bloomed in an area where the water is warmed, whereas people used to study their behavior a lot, now they're looking at them with climate change and pollution in mind because they're blooming a bit in areas where there's pollution, and sometimes, not always, but sudden appearances of jellyfish schools can be indicative of a biodiversity collapse as these jellies survive in place of other animals that are no longer there. So this is Aurelia, and it's a genus of moon jelly, named for its profile and occasional glow in certain lights. They're ubiquitous in aquariums where jellyfishes are displayed, and they're very easy to obtain, which is one reason why we have specimens in lab. They're identifiable by its 4 horseshoe-shaped gonads. It has little locomotive capability. It's flat. It drifts with currents with little vertical migration in the water column. It captures other jellyfish, very small, very, very small fish, plankton, little arthropods, and it will contract its bell and tentacles and bring that food item into its gastrovascular cavity for what is probably a pretty slow digestion. It's a pretty shallow cavity. These have a weak sting, and they're eaten by ocean sunfish, sea turtles, other jellies, and a few fish that are considered herbivorous, or at least they used to be until they were seen eating jellyfishes. So I guess they're not strict herbivores. So a persistent problem on the marine conservation front. Predators of jellyfishes, which include leatherback sea turtles, mistake plastic shopping bags for them and ingest them, and that can cause a lot of problems for them. So this is another cnidarian, Obelia. It's a genus. That's a cnidarian that exists in alternate stages of stationary polyp and free-swimming medusa. So this hydroid is asexually reproducing, and it's a colony. So all of the polyps are usually the same sex, either male or female. So you can have a male or a female hydroid colony. So this whole colony here is either male or female. I don't know how to tell the difference. Most hydrozoans, such as Obelia, are free swimming as adults, and they use the hydroid colony strategy to reproduce. So this hydroid would be rooted onto some form of substrate. The stocky, bulky, solid portion that's bright pink is composed of material called cenosarc. And that tissue is kept upright and organized by the protective parasarc, which here it looks like an expanded translucent envelope. The most animal-like parts of the colony are the gonozoids and gastrozoids, which, true to their root words, function during reproduction and feeding respectively. The breeding and feeding buds are easy to distinguish. The feeding bud has tentacles and it captures food particles like an anemone would. Medusas are released from the gonozoids and they distribute, mature, and spawn as a way to reproduce sexually, which is how genetic diversity is maintained. Physalia is an open water predatory cnidarian that floats via a gas-filled bladder, sometimes called a sail. The common name Man-O-War was issued because its sail resembled a type of British naval ship by that name, Man-O-War. Physalia is a member of Cnidaria. It's a hydrozoan, but it is not a true jellyfish. It is considered to be a siphonophore, which is distinct from jellyfish. It has a small siphon that it can use to have a little bit of autonomy over where it goes. It can produce a small burst of water to go in one direction or another. It is largely a drifter, though. But here's the big difference. True jellyfish are individual organisms with their own genome. Siphonophores, like this, are colonial entities. A single floating Physalia contains specialized zooids that are functionally distinct. It's similar to a hydroid colony in that it contains reproductive buds and feeding buds. So these deliver a painful sting. That's what they're best known for. Topical application of vinegar or diluted ammonia are usually effective remedies to stop the pain. So that is the end of the specimens for Lab 8 that we would have looked at in full. Be sure to check out the other presentations that include the slides associated with some of these, like the hydroid and the sponge.